The study of opioid addiction is uniquely challenging in that in a traditional genetic study, you would look at people who have a disease and those who don't. When it comes to a study of substance use, you really want to compare it to people who use a substance versus those who use a substance and become addicted. And so that makes it a little bit more difficult for standard genetic studies. And so the standard approach of collecting two groups and looking for differences has yet to be effective. People are certainly working on improving those studies, but we're not there yet. And so we took an alternative strategy. Rather than looking at the genetic variants themselves, we look at how the genome functions. So you can think of genetic readouts, like how much RNA is produced would be one readout of how the genome is working. Another is how the genome is actually folded and structured in cells. That is a readout of how it's functioning. And so we took a study of this structural changes, looking at individuals who died of an opioid overdose compared to some healthy controls, and looked for changes in the structure of the genome in the brain. And that revealed some interesting, unexpected findings. I think we had thought sort of naively that people who were exposed to opioids would have a pretty clear signature of change in the brain. You'd think that right after an opioid overdose, perhaps you'd see changes in the genes that express opioid receptors or some sort of larger, more ubiquitous change, common change. But that's not what we saw. It was quite heterogeneous, very different, very highly variable from person to person. And so that sort of confounded us at first. But what we ended up finding is that the changes that we observed, even though they were individual specific, they tended to occur in a similar area of the genome. So each little change was different from person to person, but they converged on specific genes. And so that allowed us to think about how those genes are important in the course of the disease, but also changes the way I think about these types of studies, which are called epigenetic studies, and how we might change the analysis approach to get new insights.